So you carry a burden of deleterious mutations which you picked up from your mother and your father, and there's a certain legacy load which came in from their ancestors, and so on and so forth. These are imposed mutations which are typically recessive and typically have very weak negative effects on your phenotype. So as a consequence, they sort of build up and up and up, in the absence of strong purifying selection. And just to make it clear, this is since the Industrial Revolution. The severity of selection has significantly decreased from 40% child mortality to about 1% today. So look at the probability of a randomly selected individual being able to fully participate in the reproduction of the next generation. And to estimate this, we have a parameter that was developed by a very brilliant researcher called Motz Henneberg, who is an anthropologist formerly based in Australia, now in Switzerland. He developed a measure called the Index of Biological State. 100% on the Index of Biological State, and that means that 100% of individuals can in theory fully contribute to the reproduction of the next generation. Now nobody has 100%, but a lot of modern populations come very close. They have values in the high 90s. So in the case of Britain today, the value is 0.98. That means that all else being equal, a randomly selected British person today has a 98% chance of being able to fully participate in the reproduction of the next generation. Historically, the index of biological state, going back now to 3,000 years, was 30%. So that means a randomly selected individual had a 30% chance of being able to fully participate in the reproduction of the next generation. It's the probability of making it to a point at which they can reproduce. So you think of it like a hurdling model. They have to get over various hurdles. They have to get past very high infant mortality, very high prenatal and prenatal mortality, very high childhood mortality, which is as high as 50% historically in places like Sweden and France in the 17th and 16th centuries. You have a lot of mortality due to violence, for example, a lot of mortality due to disease. A lot of these people, a lot of the fraction that actually made it through the selective filters would have had something wrong with them, which would have precluded them from being able to procreate. They would have been sexually selected against. So when you add up all of these filters, you can estimate the likelihood of those individuals being able to participate. Now, in reality, having an index of biological state value of, say, 0.98 does not mean that 98% of your population are going to fully participate in the reproduction of the next generation. We know that in reality 20% of the population of Western countries go childless. So it's simply telling you that all else being equal, setting aside lifestyle choices, setting aside the Easterlin hypothesis, which is the idea of trading fertility against opportunities for increased exposure to education or earning power or what have you. Taking those things out of the equation, we should be in a situation where the vast majority of people can fully participate in reproduction. The reality is it's about 80% of the population. Historically, that is still astonishingly high. Historically, 40% would be normal. So this is a considerable rise in genes which would have been selected out before the Industrial Revolution under conditions of stronger purifying or negative selection. So this is specifically selection which weeds out deleterious variants. Now, the other thing we have to keep in mind here is a given allele which causes some minor medical abnormality or loss of function or causes some kind of impairment like, say, myopia or asthma, or even causes a disease like type 1 or type 2 diabetes or even cancer in late life. The costs to having these mutations are very different today to what would have been the case in the Middle Ages, where being an asthmatic child would probably have put you at a very, very significant fitness disadvantage, given the amount of physical activity that was needed and given the absence of any kind of prophylactic medications to deal with asthma, like salbutamol sulfate and clinomodulate and these other medical miracle drugs, these steroids, which are used to treat asthma. The same with diabetes. A mutation which makes you diabetic today can be treated quite easily, palliatively, through insulin and dietary regulation. Historically, it would have been impossible to treat, and as a result, nobody even knew what the condition was. Your mortality rate would have been very high. So the cost of these disease-causing mutations has reduced. That's the reason why they are accumulating within our population. Don't take my word for it, this has been pretty much the mainstream position in human population genetics going back to the 1950s of the work of Hermann Muller, who was the first person to propose that mutations should be accumulating gradually with a fitness loss per generation of about 1%. This was reiterated, Haldane restated it, Dobzhansky restated it, Kimura, the originator of molecular neutralism, restated this, Hamilton restated this in a very famous essay which was published in the second edition of Narrow Roads to Gentlemen called The Hospitals Are Coming. He was very pessimistic about our prospects for being able to deal with the accumulation of deleterious mutations. More recently, Michael Lynch wrote about it in a paper. And Kondratiev, who's one of the absolute titans in the field of human population genetics, wrote a brilliant book about it, which came out last year called The Crumbling Genome. And it's a very, very good read. Now the thing is that 84% of the genome relates to the mind. So consequently, if people have physical mutations, they are likely to be comorbid with mental mutations. Think of a genome like a target, and you're throwing darts at a big target. And you've got on the wall this large target. And different parts of the target have different sizes. So you have a bullseye that covers 84% of the target, and then you have the outer rings which make up the remaining 16%. So if you're blindfolded, and you throw a dart at that target, what are you more likely to hit? You are more likely to hit a gene which is involved in the maintenance, regulation, and development of some brain function. And this makes sense, because if you think about what humans are, we are a highly incerebrated species relative to other primates. We have very big heads relative to our bodies. We have big brains, even in terms of just the absolute brain mass for humans. Ignoring corrections for body size, etc., humans have extremely large brains. So it makes sense, a lot of a genome is going to be involved in brain growth, maintenance, and development. You find that as fathers age, and mothers as well, but fathers are, by far and large, the greatest source of these, what are called de novo mutations. As fathers age, they bequeath to their offspring something like 1 and a bit de novo mutations. So about 1.4 de novo mutations per year of advanced paternal age, per diploid gamete or per offspring. So at the age of 35, the average father will have conferred something like 30 to 70 new mutations to their offspring's genome, and were able to count them. So people have developed assays which allow you to actually count them. You can compare the genomes, you can do the sequencing of the offspring, you can do the sequencing of the parent, and you can work out what is present in the offspring that isn't present in a parent. And if you correlate the difference with the age of the parents, you get an idea of how the age of the parents actually affects the accumulation of mutations. So in modern Western populations, in the absence of purifying selection against these variants, which are fairly minor, these are mostly what are called synonymous variants. That's where you have a specific base pair that is switched out for another base pair. These are synonymous variants. They mostly do nothing. They are not totally benign. They do affect protein coding. They do affect fitness. This has been shown in mutation accumulation experiments with Drosophila melanogaster fruit flies, where they've looked just at the accumulation of synonymous variants, and they found that there are fitness constipation. But don't forget, that's not going to be all of the mutations that you acquire from your parents. For mothers, it's about a quarter of that. So they transmit about a quarter of those numbers of mutations per advanced year of maternal. For the longest while, people thought they just came from the fathers. But no, it turns out you get some from the mothers as well. Is advanced paternal age more dangerous than the maternal age? There's some debate about that. For some traits, possibly, like chromosomal aneuploidy, where you're talking about getting an extra chromosome 21, for example, and you have Down syndrome. But for these sort of point mutation type diseases, I think the jury is still out on whether maternal age versus paternal age. It's not good to have either. The molecular etiology of autism is interesting because there are other things as well which are going on there too. So, yes, there have been some studies showing that autism is quite robustly associated with paternal age. And as a consequence, that suggests that, yes, some of the factors which cause autism are related to having large burdens of these synonymous point variants. However, there is also literature showing that autism is associated with copy number variation. And copy number variation is basically a bigger form of mutation. Copy number variation tends to be more harmful. It's much rare. These are rare variants, whereas these synonymous mutations is what are called common variants. Rare variants play a role in autism and also intellectual disability more generally. Some of these rare variants are characterized by not one single base pair changing, but by large numbers of base pairs either being deleted or being added to the genotypes. You get chunks of a genome which are duplicated. And sometimes you get the situation where you have a sequence like this, where you have a base sequence here, and you have another base sequence here, and they get flipped. So it's called an inversion. So you get inversions, you get deletions, you get additions, and you get substitutions as well, where entire chunks of a genome can be replaced with other chunks of the genome. And that's what CNV are. That's what copy number variation is. And copy number variation is bad news. And it turns out for autism, copy number variation also plays a role. But here's an interesting fact. Copy number variation is not, to the best of our knowledge, associated with advanced paternal age. It seems to be a kind of additional thing that happens in addition to you getting all these point mutations, including some bad ones like insertion, deletion, point mutations, which might come in as a function of paternal age. Most of those mutations are going to be benign, more or less. They're going to be synonymous variants. In addition to that, you might get these other mutations, legacy mutations, coming in, which are either legacy mutations or de novo mutations, which arise spontaneously independently of advanced paternal age and will interact with one another to produce this perfect storm of genetic dysfunction.